Okay, so thank you so much for the introduction, and I'd like to thank Natalie and James for organizing and for inviting me, to giving me the opportunity to share my work. What I would like to tell you about today is about an organoid model that we developed in the lab of this region of the brain called the choroid plexus, and I hope I can convince you today that this region is actually quite interesting. So a little bit of introduction. So the choroid plexus is a secretory tissue located in the ventricular system in the brain. So here you can see in blue the ventricular system, which is composed of lateral ventricles, third ventricle and fourth ventricle, which are interconnected with each other, and they are filled with the cerebrospinal fluid. In red, you can see the location of the choroid plexus, or the different choroid plexi in the lateral ventricle, so emerging in the lumen of the ventricle. And here you can see a section of actually a human fetal brain with indicated the choroid plexus there. And you can see the morphology is actually a pretty convoluted tissue, and this tissue is composed by an epithelial component, and which surround a connective tissue and fenestrated capillaries. The cell of the choroid plexus are highly polarized, so they have an apical side with microvilli, which face the lumen of the ventricle, and the basal side facing the vasculature. And these cells are also tightly packed with tight junction. So there are several functions of the choroid plexus-CSF system, and I will talk about the main function today. The main function is probably the secretion of the cerebrospinal fluid. So this tissue is a highly secretory machine, and the CSF not only provides a mechanical support in the brain, which surrounds the whole central nervous system, and but is also a supply of vital nutrients such as vitamin C and folate, as well as hormones and signaling molecules. And these signaling factors are transported in the CSF, and they can affect the neuroepithelial stem cell, for example, promoting their proliferation. The other important function is the interface between blood and brain. So the choroid plexus, it's a tight barrier and controls the entry of drugs and toxic compounds in the brain. But there's other function. There's several other functions, such as the clearance of amyloid, which has been implicated in aging and in Alzheimer's disease and other neurodegenerative diseases. There's a neuroimmune modulation role of the choroid plexus because it's lined with macrophages and immune cells, and it is also a point of entry of several pathogens in the brain. And it has been recently implicated in the circadian regulation of the brain. So especially during sleep, there seems to be changes in flow in the CSF. As I mentioned, the choroid plexus forms a barrier, which is called the BLOOD-CSF barrier. And there's other barriers in our brains. The most studied is probably the blood-brain barrier, which is a complex barrier to study because it's composed by multiple cell types, but mainly formed by the tight junction between the endothelial cells. So whether the choroid plexus barrier is formed by the epithelial cells of the choroid plexus itself, and it actually forms earlier in development compared to the blood-brain barrier. So there's several limitations, though, in the study of this tissue and in the study of CSF in human especially. So the anatomical localization makes it challenging to dissect because it's located deeply in the brain. There's challenges to isolate pure CSF without blood contaminants when we are studying CSF in vivo, because often we have a ton of blood proteins mixed in with the CSF. And there's ethical and technical challenges related to the collection of human biopsies, especially during development. So we were interested in the lab in study this region of the brain for its function, and therefore we decided to generate an enriched cortical organoid with this region of the brain. So in the lab, we routinely generate cerebral organoids to study brain development and evolution. We generate them using pluripotent stem cell. We aggregate them forming embryoid bodies. We then promote the formation of neuroectoderm, and then we embed them in matrigel, which allows the formation of this neural tube-like structure. Then we basically incubate the organoids in a very essential media, which allowed the spontaneous formation of this cytoarchitecture, which resemble stages of fetal brain cortex. And we can properly see the development of ventricular zone, subventricular zone, and cortical plate where the neurons align. And this region can become, this organoid region can become quite enlarged. And we can see specific markers of neurons and neuronal progenitors, which really resemble the cytoarchitecture of this tissue. But since we are using, we're not differentiating this organ, we're not patterning them with specific molecules, we often, we occasionally see the development of other brain region. And sometimes, since we're forming mostly dorsal telencephalon and dorsal telecenphalic identities, we can see also development of choroid plexus, which is adjacent to it. And here you can see it in green, stained by the marker TTR. You can actually see that the morphology is very different from these neuroepithelial buds that we normally see in the organoids. So, but how does this tissue develop in vivo in humans? So there's not much known actually about development of choroid plexus in human. But, and this is a very simplified schematic, but we know that it starts, it emerges from gestational week 7. You can see in red the choroid plexus primordium adjacent to this region called the cortical hem. And we know that important for this, its development is a local gradient of BMP signals and also Wnt signals, which are secreted by the cortical hem. So it is not in particular that BMP4 is sufficient to induce choroid plexus epithelial fate from embryonic stem cells. So we basically took our protocol to generate cortical organoids, and we added a pulse of BMP4 and CHIR, which is a Wnt signaling activator molecule, to basically push the development and the dorsalization of our organoid to form choroid plexus. And indeed, after only 4 days of treatment, we can see an elongation of this neuroepithelial bud, which is consistent with basically in the differentiation of choroid plexus identities. So we know that the choroid plexus derives from the neuroepithelium itself, which is a pseudostratified epithelium, and then it becomes more and more elongated and a thin line of cuboidal epithelial cells. And we can see exactly the same thing in our cortical organoids that are patterned for this using this molecule. So you can see here a series of histological sections of control, untreated, and treated organoids, which progressively develop this identity, and they are positive for this marker TTR. By day 40, these organoids are completely enriched in this epithelial tissue. And we basically, when we compare them with the section of human fetal choroid plexus and mouse embryonic choroid plexus, we can see that there's a resemblance with the human fetal choroid plexus. And also we can see other cell types such as stromal cells, and they are very abundant in human choroid plexus compared to other animal models such as rodents. We performed some ultra characterization of this tissue with EM, and we can see the formation of tight junction of microvilli and cilia, as well as multivesicular bodies and extracellular vesicles, which are indicative of a secretory tissue. So then to better characterize this tissue, we decided to perform single-cell RNA-seq, and we wanted to really look at the populations that we generate and they are present in our organoids. So what we did was to compare the choroid plexus organoid at different stages with the telencephalic control organoid. So here you see a UMAP plot of the telencephalic untreated organoid, and you can see, as expected, that we see that we have a neuronal progenitor and immature neurons cluster, as well as neuronal clusters in this organoids. And here you see the different choroid plexus organoids at different time points, and these are also from different batches. And you can see that there is a progressive enrichment of immature choroid plexus cluster, and this is based on differential gene expression in this cluster, as well as known markers of these cells. And we can see this sort of orange cluster, that's the immature choroid plexus and hem, and then progressively this sort of olive green cluster here. I don't have my pointer, but you can see that there is a maturation of the choroid plexus, as well as the stromal cluster, which is this pink cluster below. So we can see specific markers enriched in this cluster, such as CLIC6 and HTR2C, which are choroid plexus marker of mature tissue. So then we decided to compare this data set of single-cell RNA-seq with the published data set of single-cell RNA-seq from human and from mouse choroid plexus. So we can see that indeed our single-cell RNA-seq data set seems to cluster unbiasedly with the human choroid plexus, and this is true for both the mature choroid plexus and the stromal tissue, and is clustering closer to human than compared to mouse as well. So basically we seem to recapitulate the transcriptomic signature of human developing choroid plexus. One striking feature that we observed in this organoid was the generation of this fluid-filled cyst, or compartments filled with fluid. And we noticed that the choroid plexus itself was lining the barrier, basically the perimeter of the cyst, and it seemed to be, the matrigel seemed to be important for the correct localization and polarity of these cells. So we looked at also how the specific markers such as CLIC6 and aquaporin were localized, and we could see the correct localization in the tissue. So then we wanted to test whether this organoid could be actually forming a barrier, whether the choroid plexus in our system was actually forming this barrier. So we looked at cloudings, specific cloudings expressed in the choroid plexus and also in the blood-brain barrier, and we can see indeed expression of clouding 1, 3, clouding 5 as well, and others. And this is true also by single-cell RNA-seq. So we decided to first do a very simple experiment and to add fluorescently labeled dyes, dextrans, with different molecular weight to really test the entry of these dextrans, which are inert molecules, inside of the organoid fluid, which we can extract and really analyze. We can extract several milliliters of this fluid from multiple organoids. So we just simply put the dye in the media, and you can see here the media is blue, but the actual inside of the organoid doesn't allow, there's no entry of this dye, so we don't detect barely any of this. So this suggests that we have a simple barrier forming, but we wanted to really test this barrier with a more stringent sort of small molecules. So we decided to look at native state of these two very similar small molecules that have opposite permeabilities, so levodopa and dopamine. So it is known that dopamine cannot cross the barrier, the blood-brain barrier, as well as the blood-CSF barrier. And levodopa, which is a precursor of dopamine, can cross. And in fact, it is given to a patient with Parkinson's disease to replace the dopamine loss. Because it can then be converted into dopamine once it enters the brain. So we decided to add both of these molecules in our system and then basically detect after 2 hours the presence of them in the media and inside of the organoid by NMR. So what you see here is the NMR spectra of control levodopa and dopamine after 2 hours inside of the organoid. You can see in the control there's no molecule, so we don't detect any peaks. And then in the levodopa, we can see these very clear peaks. And in the dopamine, the specific peaks of dopamine are not detectable or very, very low. And we can quantify this by basically, we can see that in the media, after 2 hours, both molecules are indeed present, but inside of the organoid, only levodopa is detectable and barely any dopamine. We can detect barely any dopamine. So this is possible because it's known that LAT1, which is the transporter of levodopa, should be expressed to allow the transport of levodopa indeed, and we can see it in our tissue, as well as we can see other efflux transporter and other important transporter that are characteristic of this barrier. So then we wanted to see whether we can also recapitulate other known drugs that are known to cross or not cross this barrier. So we looked into bupropion, which is an antidepressant that can cross this barrier, whether methotrexate is a chemotherapeutic agent that does not cross, and we can indeed recapitulate this, but we can also see that we can really correlate the level of permeability with what's reported in vivo with a slope of 1, so not only qualitatively but also quantitatively, like the dose can recapitulate what's reported in vivo. So then we wanted to answer the question whether we can also look at kinetics, because often clinical trial can fail because of there's a problem with pharmacokinetics or abnormal accumulation of the drug, or the drug does not cross the barrier. So we decided to look at this molecule called BIA, which caused unfortunately a failure in this clinical trial, which left 1 patient brain dead and 5 patients were critically ill. So what went wrong in this trial? So the 6 patients in this group were in the escalating dose group. The symptoms that this patient manifested appeared after days after the initial dose, and they were still there even after the level of the drugs in the blood were negligible. But importantly, none of this toxicity was predicted in the animal models because there was not a human preclinical model to look at this. So we tested in our human choroid plexus organoid, and we can see that the drug can indeed cross this barrier. But what we did was to look at the kinetics of this drug, comparing it to bupropion, which is a safe compound. So we can see here that the bupropion basically returns to base level after 2 days, whether this drug BIA keeps on accumulating inside of the organoid even after 72 hours. So together with other toxic effects that have been reported for this drug, these organoid data support the theory and perhaps would have predicted also an accumulation of this drug inside of the brain. And there's still more work to do on this, but this perhaps would have raised some flags. Another drug that we were interested in was this novel compound, which is, we don't have yet human data on it, but the clinical trial have started. We only have data in mouse, and this is cefin-1, which is a proteostatic agent. And so we can see that indeed this drug can cross this barrier. But when we look at the level, we can see that indeed the organoid data seem to point to a lower permeability in humans. So perhaps suggesting that a higher dose of this drug may be needed to reach the same brain concentration in human compared to mouse. And this is possible because mouse, for example, have different level of expression of certain transporter along this barrier compared to human. But what we were most interested is really the composition and what was this fluid inside of this organoid. We were really curious to see and to answer the question, is this real CSF? So since we can extract loads of it, we performed this experiment by mass spec analysis, in which we compare different organoids, different batches and different cell lines. And we compare it with the in vivo CSF taken from human adult mouse embryonic CSF and also bovine fetal CSF. And we can see a large overlap of the protein detected in the organoid with the protein detected in vivo CSF. And when we looked at the enrichment analysis of the organoid CSF, we can see that there is indeed categories enriched, such as extracellular space, exocytosis, but also regulation of IGF, which is a signaling molecules present in CSF and in developing CSF. So when we look at the most abundant protein present in our organoid, we can see that they all also are shared with in vivo CSF and not with the medium. And here in red, these red arrows indicate proteins that are known biological biomarkers for various diseases, which include Alzheimer's disease, but also multiple sclerosis and traumatic brain injury. So we can detect more than 50 biomarkers in this fluid. So then we wanted to look, because we knew that in vivo dataset published of CSF, there's a lot of blood contaminants. So since we analyzed also human CSF from in vivo, we decided to look at what we can't detect in our organoid, but is present in this dataset. And indeed, we can see that the most enriched component of this subset of protein is blood microparticle. We can detect hemoglobin and aptoglobin. So basically, this means that our system, which lacks of vasculature, is able to really secrete what is a more pure CSF fluid, perhaps. There's pro and cons about this, because obviously there's no vasculature in one way, but in the other way, we have the advantage of look at the system in isolation. We knew that organoids mature over time, and therefore we wanted to ask whether also the CSF can mature over time. And we compare our CSF dataset with the published dataset of pediatric, embryonic, and adult human CSF. And we can see certain overlap with this dataset, which allow us to basically almost split our list of proteins in proteins that were more abundant early, up to day 58, with proteins that were more abundant later. And we can see indeed that the proteins that were more abundant earlier were shared with embryonic CSF, and some of them were enriched, and we detected lots of proteins such as collagens and extracellular matrix proteins early on, as well as alpha-fetoprotein. And then later on, we see an enrichment more in lipid proteins, such as ApoE and apolipoproteins, clustering, and this is also true when we look at the more mature organoids. We can see an abundance of these lipid droplets in the tissue. Finally, we were interested in looking at different cell types in the choroid plexus population, because when we looked at our single-cell RNA-seq data in the mature choroid plexus cluster, certain biomarkers were homogeneously present in this cluster, but others were more less consistent, they were more dishomogeneous. Therefore, we decided to perform subclustering of this population, and we identified indeed 4 big populations in this cluster based on differential gene expression. In particular, we found this dark cell cluster, which has been reported actually in the literature in very old papers from the 70s. There were some electron microscopy data of these cells, which are highly enriched in mitochondria, but they express less ciliary assembly protein, whether the light cell are exactly the opposite. So they express less mitochondria and have more ciliary protein. So we actually looked at the EM data, and we can see the presence of these alternating dark and light cells actually by EM. And you can see here this 2 dark cell, and in the middle this light cell with a ciliary body. And then we look at the protein, we can actually see a number of genes enriched in these clusters. Another population that we detected was these myoepithelial cells, which expressed markers of epithelial choroid plexus markers, but also markers of actin and cytoskeletal contractile fibers. And we can actually see the presence of these myoepithelial cells in our tissue. They're quite sparse, but we can still see them. And these cells have been interestingly found in secretory glands, and they seem to promote actually the contraction, and which promotes the secretion of the CSF or whatever secretory fluid in case. And then what we did was actually to look at what was secreted by the different cell types, because we were able to merge the transcriptomic data with the mass spec data. So the single cell RNA-seq with the transcriptomic data. So with this, I hope I could convince you that this tissue is actually pretty cool, and we can generate this tissue by basically patterning our organoids. we can replicate two of the main function of this tissue, which is the permeability and the barrier function. We can recapitulate the selectivity of these small molecules, but we can also generate this human CSF-like fluid in vitro. And this has been recently published, so if you're interested, there is the link to the paper. And with this, I would like to thank also my lab, Madeline, for the constant support, and my collaborators. And if you have any question, I'm happy to answer, or you can also email me. Thank you.
Thank you, Laura. That's some incredible work. Really, and I've read the paper, and it's a really stunning paper to read, and I really recommend it. So if you can post questions in the group chat, please.
Should I stop sharing?
Yeah, if you could stop sharing, that would be great. We haven't got too long for questions. I have a quick one. So you really nicely articulated the advantage of using this as a method of screening the permeability. And then you also hinted about the sort of biomarkers which you picked up. So could you envisage this would be used to look at patient-specific choroid plexus and then identify biomarkers of disease?
Yeah, definitely. That's an avenue that we are really interested in looking at. In particular, we are now starting to collaborate with somebody that works on multiple sclerosis. So we can look at specific marker by adding the sera from patient and just see what the choroid plexus secretes in terms of biomarkers. Yeah. But another thing you can imagine doing is obviously also generating organoids with mutations and look at the various specific biomarkers from Alzheimer's disease mutations or Parkinson's disease mutation, and see whether you can detect them early on. That's definitely interesting.
I think that could be really powerful. We have a question from Ritu, who says, beautiful work. Have you done comparisons of the CSF secretion differences between organoids, and what level of variation do you expect?
Sorry, do you mean between organoids at different ages or different?
It's not clear. I think probably maybe within a batch or batch to batch.
Batch to batch. I have to say we haven't looked exactly batch to batch. Like within the same batch, we haven't looked, but we have looked at differences between batches, and they're pretty similar. We can detect the most abundant protein. We have done this in the paper. Actually, I think we show that the most abundant protein seem to be there between batches, so there doesn't seem to be a huge variation in the most abundant proteins. However, we see differences in less abundant proteins, which are harder to detect as well. Yeah.
Okay, sure. Thank you. We have a question from Hope, who says, from what I caught, BMP plays a role in allowing the choroid plexus to grow. But can you describe the BMP and exactly how it does this?
How it's actually involved in the promotion of the tissue.
Can you explain what BMP is and how it's used as a?
So BMP is a secreted morphogen which acts on a receptor, which is there's a different receptor, but I think the receptor is the BMP receptor 2, if I'm not. Hopefully I'm not wrong. And it basically, initiate like transcriptory program that allows the development of choroid plexus. But there's also other factors. I really simplified a lot the background on the development because it's much more complex than that. But BMP4 seems to be essential because the knockout of the receptor, if I remember correctly, does not allow the formation of choroid plexus in animal models. So you don't have actually generation of choroid plexus if you knock out its receptor. So it seems to be pretty important. And also alone BMP4 seems to be really key of just alone this molecule to promote epithelial choroid plexus fate from stem cells.
Sure. I think we have time for maybe one more question. So, okay. So we have a question from Akash, who said, why was the total set of biomarkers larger for the organoids than for in vivo? Is that the case?
The total set of biomarkers larger for, so we do detect different numbers of biomarkers because we haven't done a proper quantitative study. This is more the overall, what we detect is a semi-quantitative. We look at the MPI level. So we do select the one that express above a certain level. So they have an MPI above 1, and we detect in at least 3 organoids or 2 organoids from each data set. But yeah, so there's differences obviously in that. Yeah, perhaps there's more. Yeah, there's more less abundant protein in the human CSF that we see. Yeah.
Okay. Maybe just one quick question, one quick answer, if that's okay. So Prospero says, have you experienced the presence of the CSF fluid even without using the BMP4? So maybe in the whole brain cerebral organoids, do you see that appearing?
So yeah, we actually, initially, we did see the development of spontaneous choroid plexus in our cerebral organoid, as I showed. And we do see sometimes see the development of this cyst also in the untreated organoids because they have some choroid plexus, so they normally develop some of this cerebral spinal fluid containing cyst. And we have compared actually the fluid from organoid that have some cortical tissue as well as choroid plexus, and we do see more neural secretory marker from neural tissue, so we can detect other markers coming from the brain as well, because obviously the surrounding environment does affect the composition of the CSF. So we do see it sometimes, but we definitely enrich more with this protocol. It's more reproducible.
All right, thank you. There's more questions rolling in, but I think we have to move on now. So
thank you
maybe
very much.
Thank you. If people are really interested, maybe they can contact Laura. But thank you for a wonderful talk. And I'm going to hand over now to Natalie for who's going to introduce our second speaker.